Good evening. It's 8 o'clock, and welcome to today's episode of Marvelous Medicine, which is on cutting edge research in inflammatory bowel disease. Our speaker today is our newly, anointed Dr.Subashree Rajaram. She's a postdoctoral researcher at the University College of Dublin. Subhasree did her BTech in genetic engineering from SRM University, India. She went on to do her MSc molecular cell biology at the University College Cork. Like I mentioned, she just completed her PhD in biochemistry and cell biology from APC Microbiome and University College Cork, Ireland. During the past few years, Subhasree has done a lot of work on cytokine synergism in inflammatory bowel diseases and is currently developing new molecular targets for triple negative breast cancer. Subhasree is a semi-classical dancer and a henna artist. Thank you, Subhasree, for readily accepting to present your work for Marvelous Medicine.
And thank you, Dr. Vidya. And I would also like to thank Radhakrishnan sir to giving me this opportunity, and Dr. Basumani for accepting to moderate this session.
So, like you mentioned, moderating the session will be Dr. P. Basumani, a friend and an ex-colleague. Dr. Basumani is a consultant medical gastroenterologist at Apollo Hospitals, Chennai. He did his MBBS from Stanley Medical College and MD Internal Medicine from Chipma, Pondicherry. So I know him from my postgraduate days. He went to UK and completed his MRCP and received his certificate of completion of specialist training in gastroenterology. He was subsequently awarded the Fellowship of the Royal College of Physicians. After working in NHS for almost 20 years, he moved to Chennai. Dr. Basumani is a clinician, academician, and a great orator in both English and Tamil. Maybe he speaks Hindi as well, I don't know, but I've heard him speak both to lay people as well as to professionals in English and Tamil. He's a wonderful singer and a movie buff. Thanks, Basumani, for accepting to moderate the session at such short notice. Over to you, Subhasree.
So thank you. So as I said, today's session would be for cutting edge research, and particularly I'm going to talk about my research that was mainly on cytokine synergism in inflammatory bowel disease. So throughout this, in this presentation, I'll be talking about a few technologies that we have used to understand the cytokine synergism in IBD, as well as particular results from a paper that we had published last year in Cell Death and Disease. So I'll talk about the results from that paper and the technologies as well. So just to say, sorry, I'm a PhD, and this is my first talk to the clinicians. So I'm still new to this field. I'm still learning a lot, and I'm still hoping to learn from this as well, a lot about IBD and more on other diseases as well. So I am from Ireland. Ireland is like a small country just beside the United Kingdom, and it's very green and very beautiful. And I did my research in a city called Cork, which is very vibrant in colors. It's small, but it's a valley city in Ireland, and it's very beautiful. And that's the college that I was working in, UCC. It looks more like a Hogwarts from Harry Potter. It has that kind of a feel, and it's very nice. So the institute associated with the UCC where I was working, it's called the APC Microbiome Ireland. They are leading in microbiology research in the institute. And we have a lot of themes that we look into. So the research themes are microbes to molecules, diet and microbes, studying about how our diets affect the microbiota in the gut. And there is also a cutting edge research investing gut-brain microbial axis and also the host microbe interactions. And this is where my project falls into, and particularly in the research of IBD. So what is, let's see, what is IBD, or the inflammatory bowel diseases? So they are chronic relapsing inflammatory disease of the gut. They're of two major forms. One is the ulcerative colitis, and the other is Crohn's disease. So in ulcerative colitis, what we see the inflammation throughout in the colon and the rectal region, and the inflammation is seen as continuous inflammation. and it affects only the inner layer of the bowel wall. Whereas in Crohn's disease, we see that the inflammation is very patchy and it's seen throughout the gut, and the inflammation affects all the layers of the bowel wall, and it's more severe. So the epidemiology of IBD, it's more prevalent in the Western countries compared to the Eastern, but the incidence is rising fast in the Eastern countries as well. So if you see, the Europe has, North America has the highest incidence rate of 0.5% of the population, whereas in Europe it's 0.3. And it's been steadily increasing. And if you see the number of cases, it's almost 85.1% of cases increased from 1990s to the 2017s. And in India also, if you see for this registry that I found, there are about 364 patients that have been registered, and that's the demographics spread across various parts of India. So the symptoms of IBD includes of loss of appetite, the blood in your stools, fatigues, fever, mouth sores, weight loss, and diarrhea. Now, causes of IBD, there's no one particular cause for the IBD, but it's an interplay between a lot of factors. And these factors include genetic susceptibility, whereas the presence or absence of a particular gene, example, NOT2 or MUC2 gene, then the reduced diversity of gut microbiota in the gut, environmental triggers, and epithelial and mucosal immune dysfunction. Now, what are these challenges in IBD research? According to Crohn's Colitis Foundation in 2019, there were five areas where there is unmet clinical needs that has to be compensated for. And these areas include preclinical human IBD mechanisms, the environmental triggers, novel technologies, precision medicine, and pragmatic clinical research. Now, I fall in the category working on the preclinical human IBD mechanism. This is done mainly to understand how, what's the mechanism in a cellular level, how the mechanisms in the cellular level affects the pathogenesis of IBD, and we take a bench to bedside approach where we do we identify the mechanism, then we evaluate preclinically on mouse models, and then we take it to the patients. So that's the approach that we are taking, and I fall into the identifying molecular mechanisms at the cellular level. So as I said, in this presentation, we particularly I'm going to focus on the epithelial and mucosal immune dysfunction factor of the pathogenesis of the IBD. And when I'm talking about the immune system, I'm going to focus more on the cytokines that is produced by the immune system. So there are various cytokines produced here in the inflammatory bowel disease. So just to go through this figure, what we see is that the gut is made up of a single layer of the epithelium cells that acts as a barrier between the between the bacteria and the underlying immune cells that are required for the immunity for us. And these immune cells, such as natural killer cells, the innate lymphoid cells, macrophages, and T cells play a very important role in the pathogenesis of IBD. And when these are activated, they release inflammatory cytokines that are called IL-12, the interleukins, such as IL-12, IL-23, IL-6, as well as interferon gamma, TNF alpha. And we have anti-inflammatory cytokines as well that is required. So that includes the interleukin 10 and TGF beta. And in this presentation, we are going to more focus on the cytokines interferon gamma and TNF alpha, and to understand more on the mechanism, how these two cytokines work, and to use how we will use the mechanism to identify other targets for inflammatory bowel diseases. So to look into the pathogenesis as to what happens in IBD, we see that the epithelial, the single layer of the epithelial cell barrier is damaged. And this damage is due to excessive cell death, either due to apoptotic cell death pathway or the necroptotic cell death pathway. And the reason for the chronicity that we see in the inflammation is because of this. There is an initial barrier disruption, which leads to regulated cell death, which will be apoptosis. But then this leads to an increase in the production of inflammatory cytokines that leads to further inflammation, injury, stress, And then there is a loop of this resulting in a very chronic inflammation. And the two cytokines, interferon gamma and TNF alpha, play a very, very high role in this pathogenesis of the IBD. So if you look into the current treatments that are there, that the clinicians usually take is a step-up approach, where for mild and moderate IBD, they look into immunosuppressants and anti-inflammatory drugs. But as for a moderate to severe IBD, we go for biologics and surgery. And we see that a lot of the biologics that are present at the moment in the market, we have the anti-TNFs here, which are the most used drugs at the moment. And then we have other drugs, but most of these drugs target the cytokines that are present in IBD, that are highly present in IBD patients. So if you look at the timeline of these biologics, we see that anti-TNF was almost identified 25 years back. So it has been in the market for about 25 years. But then only in the last decade or so, new drugs have come into market for as treatment therapeutics for inflammatory bowel diseases. And these include targeting of IL-12, 23 pathway, or the integrin pathway that is required for maintaining the epithelial barrier. And now recently enough, we have the JAK inhibitors that has come to the market. And these JAK inhibitors target these JAK Janus kinases are involved on downstream of the cytokine signaling pathway. So again, everything is targeting a cytokine pathway that is involved in IBD. But then why are we looking into new targets? What's the problem with anti-TNFs? So what we see is that only there are about 40 to 60% of the patients that do not respond to anti-TNFs. And either they do not, or they lose response over time. So initially they respond to it and they lose the response. So how do we overcome this problem? So one, as we saw before, we're targeting other signals, such as you target the other cytokines such as IL-12, IL-23, or you target targets that are downstream of these cytokine pathways. And also, we could identify there might be a presence of additional signal. For example, in other inflammatory diseases, we see that it's not just TNF alpha that is being involved. There are other cytokines that are being involved, and targeting those cytokines have given higher response than targeting just the TNF alpha. So identifying such a redundant cytokine would be of high importance in effective treatment for IBD. So that's what we as a group wanted to focus on, to find any other redundant cytokine. But there are a lot of cytokines that are produced. So how do you find what is the target? So to come for this, we thought we would focus on the two cytokines, which is interferon gamma and TNF alpha. Now, these interferon gamma is also found in higher levels in IBD patients. But then targeting of interferon gamma failed in preclinical models. And that is because in clinical trials also it failed. And that is because the interferon gamma does a lot of other functions as well. Not just, it also plays a role in homeostasis predominantly. So if we are targeting interferon gamma, it disrupts the homeostasis, which results in an exacerbation of disease rather than the decrease in the disease. But then interferon gamma and TNF alpha synergism is widely known in the field. So we thought we would look into this. We would study the synergism between these two cytokines and use this model of synergism to identify other cytokines that might be as targets for IBD. So the results that I will discuss has been published in this paper back in last year in Cell Death and Diseases. So as I said, the interferon gamma and TNF alpha synergism have been studied for a long time. The initial paper that came was back in 1983 that was looking into the synergism between these two cytokines. And now recently, even in COVID, we have seen that these two cytokines are present in the cytokine syndrome and the cytokine shock syndrome, and they are very highly produced in the COVID patients. And a lot of other, mechanisms have been studied in other diseases as well, such as diabetes, ischemia, perfusion injury, and very highly in antiviral responses. And in terms of, if we look into IBD, there are researches being done studying about the synergism, especially in the role of these two in epithelial cell death, reduction of epithelial proliferation, the loss of junction protein, which affects the which affects the epithelial barrier, and then reduction and motility and sensation in the gut, everything resulting in an increased pathogenesis of IBD. However, the mechanism behind which this synergism exactly exists has still not been yet investigated. So in this, so we thought we will look particularly in the epithelial cell death, the interferon gamma and TNF alpha synergistic effects on the epithelial cell death, and how does it affect, and how can we use this to identify other targets for IBD. So these two, we'll look into more details of these two cytokines. So the first is the interferon gamma. So interferon gamma is a T cell, Th1 cell inflammatory cytokine. So it's produced by CD4 plus and CD8 plus T cells. It has variety of functions. So it can do immunomodulatory functions. It has antiviral functions and anti-tumor activity functions, as well as homeostasis function. So if you look into the signaling, so to understand more on the mechanism, we have to understand how it signals in a cell. So to look into the signaling, we see that the binding of interferon gamma to its receptor can activate kinases downstream. So they are called the JAK1, JAK2, they are called the Janus kinases. So these Janus kinases, once activated, can activate STAT1. So this activated STAT1 is responsible to translocate to the nucleus and to transcribe genes that are involved in various functions that I just mentioned. Now, as opposed to interferon gamma, TNF alpha has a very complicated cellular signaling mechanism downstream. I will go through this very slowly in a moment. So TNF alpha is also an inflammatory cytokine and is produced by CD4 plus T cells and CD8 plus T cells, as well as macrophages. Now, even TNF alpha has a very pleiotropic function. It has both antiviral and anti-tumor activity as well. And then it does opposing cellular response, in the sense that TNF alpha is widely known to induce survival genes as well as induce cell death genes. Now we look into the signaling more closer. TNF alpha forms a lot of complexes when it's going to signal. So the first is the complex one, which is also the survival signaling complex. So the binding of TNF alpha to its receptor induces brings in a lot of proteins that attach to the receptor to form the complex one. And to note in particular of importance is this RIP1 protein, which is receptor interacting protein kinase protein. So this acts as a molecular switch between survival pathway or the cell death pathway for the TNF alpha signal. So when this RIPK1, what we call it as a process called ubiquitinated, this can induce NF-kappa B. So NF-kappa B is our main protein here. So activation of NF-kappa B is then results in the transcription to the translocation to the nucleus, and then the NF-kappa B then induces transcribes genes that are involved in inflammation, survival, or innate immunity. Now, as I said, RIP1 plays a major role here in the switch. So when RIP1 is not ubiquitinated, when these ubiquitins are removed, RIP1 then forms a complex. It's called the complex 2 with caspase 8 and FAD. Now, this complex leads to caspase 8 dependent apoptosis. So this is a complex 2. In particular, this is called complex 2A because this is a 2A is a caspase 8 dependent apoptosis. In another way where this RIP1 is also ubiquitinated, phosphorylated, in the case where RIP1 is phosphorylated, it induces RIP1 dependent caspase 8 dependent apoptosis. So there are two types of apoptosis here. One where RIP1 is cleaved and it's only caspase 8. The other where RIP1 is phosphorylated, and again it's called RIP1 dependent apoptosis. So this complex is called complex 2B. And in the case where the caspase 8 is inhibited, or the function of caspase 8 is blocked, we see something called as necroptosis. So necroptosis, this is the complex 2C, where the main thing is MLKL protein. So MLKL protein is the main modulator for the necroptotic pathway. So now in TNF alpha, we saw complex 1, which was the NF kappa B dependent pathway. And then we have complex 2, which mainly results in cell death, but then we have caspase 8 dependent cell death, as well as MLKL dependent necroptotic cell death. So to understand now between the synergism, what we did was we took an HT29 cell line model. So this is a cell line that has been used widely in studying IBD research. So this, we took it as a model for the representing the intestinal epithelial cells. So what we found was, in this model, if you see, when these cells are treated with concentration of interferon gamma or concentrations of TNF, we don't with TNF, we don't see much of cell death, that it doesn't affect the survival of the cells. But then with interferon gamma, there is an effect on the survival of the cells. But then when these two cytokines are in combination, we see that there is a drastic change in the amount of cells that have survived. So even at the concentration of 1 nanogram per ml, the cells, 10 nanogram per ml, the combination of cytokines kills the cells compared to either of the cytokines alone. And then if we see in these beautiful crystal violet picture, we see that in 72 hours, you see there are no cells in the plate at all. So you see that in the cytokines, there are very less cell death, but then when it's in combination, there's a lot of cell death that has been observed. So to understand more on what is happening behind this, why is these two cytokines kill the cells more, we try to do two screens. So one was what we call it as the kinome RNAi screen, and the other was a drug library screen. So in kinome RNAi screen, what we did, as I showed you in the signaling pathways, there are a lot of kinases that have been involved. In interferon gamma, we saw the Janus kinases. In TNF alpha, we talked about the RIP1 kinases, and there are other kinases that's been involved in TNF alpha pathway. So we wanted to see if we block either of these kinases, do that protect the cells from dying? Do they protect the cells? So that was one part of the screen. The other part, what we did was we wanted to try and repurpose drugs that have been that are available on the market. So we used about 2,700 drugs to see if they can protect the cells from the death between these two cytokines. So there are two parts. So now I'll say what we did in what is meant by the kinome RNAi screen. So RNAi screen meant that we use the siRNA technique in that screen. So what are siRNA? They're small interfering RNAs. So what do? When we give the siRNAs that has the sequences for our target mRNA sequences, complementary to our target mRNA sequences, we give this into the cells. And in the cells, there are proteins that bind to these siRNA, and this forms the RNA-induced silencing complexes. So that is called the RISC. So when in this RISC, what happens? The siRNA are usually double-stranded RNAs. Now this double-stranded is changed to a single-stranded with only one antisense strand kept to target the mRNA. So this antisense strand from the RISC binds to the mRNA, and then there is cleavage of the mRNA that happens. So when the mRNA is degraded, there is no signaling of there is no protein formed. So hence there's a reduction in protein, and then there is no signaling seen. So that's the technique of the siRNA. So we use this to target only the different kinases in our HT29 models to see which kinases protect our cells. So what we found was, we got two kinases, and these two were involved in the interferon gamma pathway. So that one was the JAK kinase 1 and JAK kinase 2. And if you see, it could, the result was very high, that they could protect almost equal to that of our controls. So it was very, so one, it was surprising for us to see, because we were expecting kinases from TNF alpha, because TNF alpha induced cell death is widely known. And then this was a very clear cut result, because the other kinases were not even in our uh, not even as our hits, did not even pass through as our hits. So JAK1 and JAK2. And the other very assuring fact that we got was when we did that drug library screen, the topmost hit was the drug baricitinib. So baricitinib is a JAK targeting drug. So it's a JAK inhibitor. And then we also had another hit, which was lower, but it was fedratinib, which was also a JAK inhibitor. So these two drugs, these two screens complemented each other, and it showed us that these two proteins played a major role in the cytokine-induced cell death in HT29 cells. So now this was a screen. So now what we have to do is we have to validate the screen results. So we always validate any results when we do a big screen. We always validate our results. So to validate the RNAi screen, what we did, rather than performing the siRNA, we performed the siRNA again, and also we did something called as the CRISPR-Cas9 knockout model. So the difference between the siRNA and knockouts are that siRNAs affects the mRNA level, but then in CRISPR-Cas9 knockout model, we disrupt the gene at the DNA level. So what are these CRISPR-Cas9? So these CRISPR-Cas9, they are called clustered regularly interspaced short palindromic repeats. So this was found by, this technique was used by Dr. Doudna and Dr. Charpentier in 2012, and they got the Nobel Prize in 2020 for this discovery. And then they used this CRISPR Cas9 knockouts are used majorly by microbes to protect themselves against viral infection. And we use that mechanism to induce genome editing in eukaryotic cells. And at the moment, this is the most widely used tools for genome editing. So we either do knockout, we can do we can introduce mutations in a gene to study about a particular mutations, and we can do a lot more genome editing work. So how does this protocol work? So we have something called as gRNA, which is the guide RNA. So the guide RNA has sequences that can target our DNA, and it also has a scaffold that can bind to Cas9. So Cas9 is a protein from the CRISPR mechanisms from the microbes. So this Cas9 is able to produce double-stranded breaks in our cells. So this guide RNA and Cas9 forms a complex. We call it as the RNP complex. And when we put this into the cells, so the guide RNA binds to the DNA, the complementary DNA strand, and then the Cas9 then creates a double-stranded break in the region of interest. So now when there is a break in the double-stranded, then there is non-homological end join repairing mechanism that is present in the cells. And then when this repair mechanism takes place, we have either we have the repair, and then we get wild type gene, we get insertion, or we get deletion, as well as frame shift. So when we are doing this, we either sometimes we can knock out the gene, we can introduce mutation, and depending on what you want to study, you can then select it. So how do we do this? So first we use as I said, the RNP complex. So we make this RNP complex. We transfect this into the cells. Now after this, we are not sure in which cells we are going to get frame shift, which cells we are going to get insertion or deletion. So to know more about it, what we do is we do something called a cell sorting. We use a FACS analysis, where in a 96 well plate, each well we send only one single cell. So there's only one single cell plated in one well. So the clones that we are going to get from the cells are going to be homologous. So then what we do after we expand these clones that we get, we then use Western blotting mechanism, or we use Sanger sequencing to identify if our gene of interest or if protein of interest is knocked out completely. So in case of knockout, in Western blot, we don't see a protein at all. And in Sanger sequencing, we can see if the mutation has been an insertion, deletion, or a frame shift mutation. So this is the CRISPR-Cas9 knockout. So we use this protocol to now knock out our two targets, which was Janus kinase and Janus kinases, and also STAT1, which was downstream of Janus kinases. So if you see we have clones. There are 2 clones for each of the knockouts. So JAK1, JAK2, and STAT1. And if you see in the combination of the 2 cytokines, we see that all these knockout clones are uh, resistant to the cell death by interferon and TNF alpha. And this is the Western blot, and you can see that there is no protein at all in these knockout zones. So for JAK1 knockouts, we don't see a JAK1. JAK2 knockouts, there's no JAK2. And then STAT1 knockouts, there's no STAT1. So this was assuring that the screen results have been translated here as well. And we found that this shows that JAK1, JAK2, STAT1, so the JAK-STAT pathway, the interferon pathway, had a major role in the cell death by interferon gamma and TNF alpha. So next, we wanted to also verify the results from the drug screening results. So baricitinib was one of the hits from the screen. So we also included other JAK inhibitors that have been that are in the market, and we use upadacitinib, BMS 911543, ruxolitinib. And as you can see, all these JAK inhibitors could rescue the cells from the cytokine-induced cell death. So if you see the blue, and this was mostly concentration dependent, but they could rescue the cell death. So this was this again proved that JAKs and STAT hold a major role in rescuing cells from interferon gamma and TNF alpha induced cell death. But when I showed you the signaling pathways, we saw that TNF alpha was one majorly inducing a lot of apoptosis and necroptosis cell death pathway. So we wanted to know, if blocking these pathway, even though they were not hits from the screen, if we block these pathways, do they rescue the cells or not? So in that effect, we did two things. One, we inhibited them using commercially available inhibitors, which will inhibit the functions of these proteins, and also we knocked out the proteins to see their effect. So first, when we inhibited these proteins using inhibitors, we saw that none of the inhibitors could rescue the cells from interferon gamma and TNF alpha induced cell death, but only baricitinib, which is the JAK-STAT inhibitor, was rescuing, but none other was rescuing. But then when we did the knockouts of the protein, what was surprising for us to find was that caspase-8 knockout. Now here, if I go back, now the ZVAT is a pan-caspase inhibitor. So ZVAT is going to inhibit the function of caspase-8, but it didn't rescue the cells from the cytokine-induced cell death. But then when we knocked out the caspase-8, that is, the caspase-8 protein is not made at all, we saw that was rescuing the cells from interferon gamma and TNF alpha dependent cell death. So that meant that the presence of caspase-8 had a role in the cell death rather than the activity of the enzymatic activity of the caspase-8. So to understand more, what we wanted to see was, in these knockout, how does the JAK-STAT pathway look? So for that, how does the activation of the JAK-STAT pathway look? So what we did, we used a method called the HTRF assay. So what this assay do is it measures the presence of phosphorylated STAT1 at this residue. So this measure says that if your STAT1 is an indirect measure of saying your STAT1 is activated or not. So if you say interferon gamma, it activates STAT1 very nicely. There's an increased phosphorylation of STAT1 in the parental line. So if you follow the red lines, they are the parental. So, and with the combination of the cytokines, we see that STAT1 is very nicely activated. But then what you see is in caspase-8 knockout lines, there is reduction in this STAT1 activation. So there is a reduction compared to the parental line. So there is reduced activation and of JAK-STAT pathway. So that said, all these results, what they say is that JAK-STAT pathway is very crucial in the interferon and TNF alpha dependent cell death. And that caspase 8 as a protein has a role in the sustenance of this JAK-STAT activation. So this is all a very nice results, but then we found all these results in a cell line. So how do we take it? If I told from the bench to bedside, how do we take it from there to a step? So to move a step forward, now what is the cutting edge research is what is called the organoid generation. So what are organoids? So organoids are basically stem cells. They're made from stem cells that are derived from patients' biopsies. So what we do is we take a patient biopsy. These are usually pinch biopsies from the colon, mostly it's colon, from colon pinch biopsies. We put it in a solution, and then we isolate we disrupt the crypts, and then we isolate the stem cells from the crypt epithelia. Now these stem cells are grown in these stem cells are grown in dishes, and then organoids are formed. So these organoids these are all stem cells. So this can be used for a lot of different to test a lot of different mechanisms, to effect of the immune signals, to test new drugs, bacteria, metabolites. So since these are derived from the patients, we think that this is more closer to the molecular signature of the patients, and hence we want to test the effect on these organoids. So this is how they look microscopically. This is from one of our lines. So you see that this is one single cell organoid, and then this is a mature organoid. When we give certain factors, we can see a lumen being formed here, and then the organoid and the other cells being formed. And then this is the mini gut culture. Now this is not mature, this is a stem cell. So we have a lot of organoids. This is just one organoid here that has been shown here. And we have a lot of organoids that is being formed. So in our research, what we did was we took the organoids and we tested for the effect of these cytokines, obviously. And then we did a time course. We looked at it at 24 hours, 72 hours, and so on. So you see that with interferon, there is a bit of a cell death that can be seen. The organoid is a bit disrupted. With TNF, the disruption is very high. It's increased. And with interferon and TNF, this is much more increased. So we wanted to see, tabulating these results, we saw that we got organoids from two different donors. So one was the healthy donors and the other was Crohn's disease patients. So we had organoids developed from these patients. And then we see that even in healthy, the interferon and TNF together can induce cell death, and in Crohn's disease organoids as well. What difference that we found was the Crohn's disease organoids from patients with Crohn's disease. We saw that they were much more sensitive to TNF treatment compared to the healthy individuals. But then when we looked into Crohn's disease organoids, JAK inhibitors, when we used the JAK inhibitors, they could rescue these organoids as well. This again states the fact that JAK inhibitor played a major role in cytokine-induced cell death. So this is just microscopical images to reiterate the same. So the cytox green is a marker for cell death, and you see in normal condition that interferon gamma and TNF induce a lot of cell death. And then with increasing concentration of the JAK inhibitors, you see lesser cell death. So overall, in summary, what our research told us was that the interferon gamma and TNF alpha synergize to kill human IEC. JAK1 and JAK2 are non-redundant kinases, and it was required for interferon gamma and TNF alpha driven epithelial cell death. And caspase 8 has a major role in this cell death mechanism. And then JAK inhibitors protect the intestinal organoids as well from interferon gamma and TNF alpha. And then cytokine-induced cell death is not apoptosis or necroptosis, which meant that there was a new cell, there might be a potential new cell death pathway enrolled, and research has to be done to identify this cell death pathway furthermore. So having said all this, so we have talked about interferon gamma and TNF alpha, but there is still questions of opening. What are the other targets that we want to look in? At the moment, there are a lot of drugs in clinical trials that include anti-integrin molecules, SNP receptor molecules. These are involved in maintaining the epithelial barrier. Then we have anti-interleukin molecules, particularly targeting anti-IL-12 and 23 and anti-IL-6. And we saw JAK and tyrosine kinase inhibitors that are downstream of cytokine pathways. And we have microbial therapies, toll-like receptor agonists, and everything. But then even with the JAK inhibitors, FDA recently, so we have 2 drugs now approved for IBD, tofacitinib and upadacitinib. But these 2 drugs are coming up with an FDA issued warnings, and that's due to adverse events seen that include increased risk of blood clots, heart related issues, and death as well. And these are the warnings is to use these inhibitors, and even in anti-IL-12 23 therapy, it's only to use for patients that are non-responsive or refractory to anti-TNFs. So again, it's only when you block TNF and it is a refraction, we are going to use other things. So we still have not seen what might be the redundant cytokines to TNF alpha in IBD. So to understand more on that, our current work, the GI team in APC, what they are currently researching is on to identify other cytokines that are redundant to TNF. So with the results that I've shown you now, so we are going to use this results as our basic. That is, we are going to use the mechanism with interferon gamma and TNF alpha cell death, as well as they also increase inflammation in the He29 cell line, which I have not shown the results today. But using these 2 phenotypes, what we are going to do is we are going to screen a lot of different ligands and see if any ligands have an effect similar to that of interferon gamma or similar to that of TNF alpha, and what is the mechanism behind it. So if we identify one such target, we are going to move to mouse trials so that we can use it as anti-TNF, the target, and think of combination therapies where anti-TNFs and this target is given as well. So that's what we are currently researching, and we have certain targets that we have identified and working on at the moment. So with that, I would like to thank definitely the Marvelous Medicine Forum and the Learning General Surgery for giving me this opportunity to share our research all the way from Ireland. And then my team, the gut inflammation team, and of course, the industrial collaborator, which is AbbVie, and definitely for the organoid research would not have been done without the patients, clinicians, and research nurses. So my thanks to them as well. So that's me. So I'd be happy to take any questions. And
Subhasree?
Yeah.
Hi, that was really marvelous. I think you lived up to the title of this series. So it makes a big difference when somebody who is fully immersed in research, they come and present their work. Yes. So your originality, enthusiasm, and mastery of the subject was quite infectious.
Thank you.
We are at the other end of the from bench side to bedside. We are at the other end of the spectrum. So inflammatory bowel disease is on the rise in India due to various factors, including the fast food. People diet drastically, make big changes in short intervals, and then disturb the microbiome. That is one aspect of it. I think we always had the genetic predisposition like Caucasians. Now that the immunological and epithelial aspect has exposed by you in such in an elegant manner. So these 3 things come to union to bring out the inflammatory bowel disease. So microbiomes, we need them as friends, but as they say, sometimes they can change to enemies. Now, the youngsters call them frenemies. A friend who can change to enemies are frenemies. That's one aspect with the environment and food. But we were benefited by your elaboration on the mucosal aspect of it. So when we started using steroids, no, they were targeting in a very shotgun manner. Then came the TNF alpha, which neutralized in the serum. Then the TNF alpha receptor etanercept came. And then we are now going down and down the inflammatory cascade. Only gut-specific interleukins like vedolizumab and anti-integrins, ustekinumab, where tuberculosis and other side effects are becoming less common. Only some nasopharyngitis, and we have one JAK inhibitor, tofactinib, available for use in clinicians. So I was always wondering, where do they act? And you really put that cascade, interferon and TNF as the first one, JAK and STAT as the next one, and then you went down to the capsids. So if it is not apoptosis and if it is not necroptosis, it's like Bahubali part 1. After that, you are leaving us in a lot of suspense. So then, what kind of ptosis is this? So I personally was benefited because we are seeing very sick patients not responding to the available monoclonals. We are really welcoming and looking forward to new one. And your double hit agent, that I really liked it. One with the TNF alpha and other with the anti-target. So that kind of we do need. That's where the personalized and precision medicine is going. Thank you for being there, doing the hard work, and giving the benefits of your research. So over to Vidya. Vidyaji, I can't see any questions in the box because I think most of your talk went over our head, I think.
That's yeah, I thought so too, but I tried to keep a lot more.
No. You did wonderful.
If you look at that paper, it's much more complex. We have done a lot more other things that I have not shown. It's a big paper, and we are doing a lot more other work, but because they are still not published, I couldn't speak about it. And with regards to your question on cell death, it was surprising to us as well. We didn't just see apoptosis and necroptosis. We also looked into the ROS-dependent cell death, which is now seen in a lot of diseases. And then there are almost ferroptosis, which is iron-related cell death, and then there is pyroptosis as well. So that is dependent on inflammasomes production and other stuff as well. So what was interesting to us as well was it was none of these cell death, and the microscopic characterization is also very different, but we are still investigating it. So it will be published soon, I hope so, which was really interesting for us. So that's where we are, and hopefully if we can find a particular mechanism by which this cell death is happening, then I think even with the anti-TNFs targeting, rather than targeting anti-TNF, which is top of the cascade, and if we can target this at the more near the more deep down, it would be much more beneficial, and that's what we are hoping for as well. So
thank you.
If we have any other medical gastroenterologists logged in, we'd like to hear from them. Dr. V. Raghavan, you have any query? So the the gauntlet falls on Radhakrishna. How much of this did you understand? No.
I'm the wrong person, Vidya. First of all, thanks, Subhasree, for accepting our request and showing us, what hard work goes on and how much brains are used in that. Because, If it goes above Basmani's head, obviously it'll go, much higher above my head. But again, behind the fruits of your research, the drugs which we use, and for me, I'm a surgeon and we do operate on Crohn's disease. For us, we are very blind to most of what you're doing. But wonderful. I mean, I really appreciate your enthusiasm in your presentation and your authority in what you're talking about. I think that shows, and your anticipation that there are much more research to come to be published, and thanks for all these efforts, and we hope to learn some more topics, and all I can say is that it's wonderful listening.
Thank you so much. No, but we should also thank clinicians, because they are the people dealing with patients to take our research to the patients, and that's what we are working for. And as I showed in that figure, it's a circle. It's never one person. It's a team of people, ultimately having only one focus, the benefit of the patient. So it's always the teamwork. And without clinicians, again, we are not able to do these organoids work and others, because we have to work in close with them, because they are diagnosing. So when we get a biopsy, it's coming from a diagnosed clinician. So we don't know what it is until we are going to work on it. So that's characterized beforehand for us. So it's again, as I said, it's a team, it's a circle. So
it's we who get all the smiles and thanks and bouquets and chocolates and stuff, but then the hard work is from you. You don't get to see the good faces, the happy faces, which we do.
Yeah, that's correct. Yeah, we always work behind the screen.
Yeah, they'll work for the Nobel Prize, Radha. So thank you, Subhasree, for making a very complicated topic, dumbing it down sufficiently that we could at least figure out what was going on, and also to cover such a huge topic in bang on time. I mean, that was really amazing that you thought you
39 minutes.
Yeah.
I was very happy about that.
Yeah. So most people are very passionate about their work, sometimes get carried away, but you did a great job, and maybe a few months down the line, once your latest work is published, we'll have you on board once again to share your exciting journey. And by then you're probably into something newer, and we'd like to hear from you. It's great to have young, passionate, hardcore researchers also on board Marvelous Medicine, because most of the time, it's more like clinician oriented. But I think it's extremely important for clinicians to know what's going on, and like Radha said, how much hard work goes in before we actually prescribe a medication. So thank you, Subhasree, once again, and thank you so much, Basmani, for agreeing to moderate this session at a really very short notice, and just for. So thank you everyone for logging in, and we'll meet again next week with another episode of Marvelous Medicine. Till then, take care and stay safe. Good night.
Thank you.
Thank you so much.